So, welcome. I will quickly introduce myself. I'm David. I'm a PhD at SITS Lab, and I might have a little bit different background than most of you because I'm originally a biologist. And after my master's here at ETH, I worked in industry, in biotechnology, as in protein engineering. And after that, I did a second master's degree, more in the field of data science. And now, after this, I'm doing my PhD at SITS Lab, working on graph-based engineering of proteins and small molecules. And the focus of this lecture will be mainly on AI models for engineering of proteins and small molecules. And so, for example, to develop new drugs or to develop new enzymes that catalyze some specific reactions. And this is an extremely dynamic field that has experienced a couple of breakthroughs in the recent years. And I will just try to give an overview over the state of the research in the field and the AI models that are used to help reach the goals of protein and small molecule engineering. So in the first part, I will quickly give a short introduction on proteins and small molecules, just that we are to explain the basics, that we are all on the same page. And then I will start talking about text-based AI models for protein engineering and small molecule engineering. And then I would like to speak also about structure-based AI models for protein engineering. But to do this, I would like to first explain the AI models that are now used to predict the structure of proteins. So you probably all have heard about AlphaFold and maybe also ESM. And yes, after this, I will explain the most common approaches for structure-based AI for protein engineering and engineering of small molecules. And then in the last part, I'll try to give an overview over the field of generative AI for de novo protein design. But this will probably be on Friday. So and if you have any questions, just raise your hand or write into the chat. So first of all, the introduction to proteins and small molecules. So small molecules are organic compounds that are small, of a low molecular weight, and usually have less than 100 atoms. So organisms produce such small molecules, for example, as metabolites or signaling molecules. For example, this cortisol here is a stress hormone and drives our metabolism. And these small molecules, they can normally easily diffuse across cell membranes, and they can interact with proteins and influence their function. And because of this, it makes sense that most drugs are small molecules because they can go into the cells and interact with proteins and change some disease pathway, for example. And the structure of these small molecules, because they are so small, can usually allows for easy synthesis in the chemical lab. So, and now proteins are larger, chain-like molecules made of a fixed set of building blocks. So they are chains, and these chains, they fold into a defined 3D structure known as the native conformation of the protein. So it's hard to see here, but this is a chain, but it's folding in 3D space. And then these chains usually fold into some kind of globular structure. So as you can see here with this approximated surface of this specific protein here. And this structure defines also the function of the protein. And these can be many things, like proteins can be enzymes, so proteins that catalyze specific chemical reactions. or they can be structural components, signaling molecules, transporters. But like in general, they are extremely important. They are like the building blocks of our cells. They play like a key role in almost every process necessary for life. So, as I said, these proteins, they are built of a fixed set of building blocks. And these building blocks are amino acids. And there are 20 standard amino acids. You can see them here. And all these amino acids have kind of a backbone. So it's an amino group and a carboxyl group. And you can always see this here. This triangle of N, C, and C is shared by all amino acids, right? So these are like the links of the chain. So if you create a chain of amino acids that folds into a protein, these triangles here that every amino acid has are like linked together to form this chain. And what differentiates the different amino acids are the side chains. So you can see, while this triangle here is identical for all amino acids, the difference is what is attached here at this middle atom here, at this C, what is called the C alpha atom. And this attachment, this side chain, is different for all amino acids and also defines like the chemical properties of these amino acids. And another thing that highlights like how important proteins are is, of course, the fact that our DNA dictates the sequence of proteins, like the sequence of amino acids in proteins, sorry. So each triplet of DNA nucleotides encodes for a specific amino acid. So our DNA is kind of read and transcribed into RNA, and this RNA is then translated into a chain of amino acids. And then once this chain of amino acid is synthesized, then it folds into a more ordered three-dimensional structure, which then defines the biological function of the protein. So how this fold looks like is entirely defined by the sequence of the amino acids and their side chains. So this folding is kind of a spontaneous process of energy minimization that is just guided by interactions between the amino acids and between their side chains, for example, like hydrophobic interactions, hydrogen bonds, or van der Waals forces. And then to describe a protein, you can like think of these three levels of structure, basically. So you have the primary structure, which is just the sequence of the amino acids in the chain. And then there's secondary structure, which are like regularly repeating local structures stabilized by hydrogen bonds. So for example, alpha helices or beta sheets. So this here would be an example for an alpha helix, and this would be a beta sheet. And then tertiary structure relates to like the spatial relationships of these secondary structures to each other. So primary structure is just which amino acid comes after which. Then this defines the secondary structures, like it forms into an alpha helix or at some point into a beta sheet. And then the third level is how these secondary structure organizations, how they fold into like a protein and how they relate to each other in three-dimensional space. And this then gives the protein its final shape. And you will very often see the proteins in this cartoon depiction. And this is just used to visualize the secondary structures like the helix and the beta sheets. And it gives a nice like overview over the structure, but it only shows like the location of the backbone of the amino acid, so of these three triangle atoms that each amino acid has. So the side chains are not visible in this. So if you would like show the side chains as well, then the protein would have a different shape, like this here on the right side. But this cartoon just shows the location of the backbone, basically. So now I will quickly talk about like how like engineering of proteins and small molecules and what the goals are of this field. So, as I told you, most drugs are small molecules, and they can bind to proteins and affect biological processes. And the chemical structure and the composition of these small molecules define their function and their binding preferences. So small molecule drugs are usually engineered to interact with some specific desired target and to bind to it, to inhibit it, and modulate some disease pathway. So the small molecule might, for example, inhibit an enzyme or block a receptor. Like you can see here, this molecule here is aspirin that binds to this enzyme cyclooxygenase, which would otherwise form prostaglandins and lead to inflammation and fever. So engineering of small molecules happens through altering the size and the composition of these organic compounds to get some desired properties and binding preferences. And then in engineering of proteins, it's all about changing the sequence of the amino acids in the chain to achieve a specific function of the resulting protein. So, for example, in the field of biocatalysis, so in the field of the research of enzymes, you could, for example, change an amino acid sequence of an enzyme to accept a new substrate. For example, here you want this substrate to be catalyzed or this to be processed by this enzyme. You might change some of these amino acids here so that this substrate can enter the active site or this pocket in the protein structure. And if you think about therapeutics, a very common example would be that you change the sequence of an antibody so that it can, for example, bind to SARS-CoV-2 spike protein. And there are a couple of strategies to do protein engineering. So there's rational design, which is just you use very detailed knowledge that you have about your protein or your system to make desired changes. So again, in this picture, you might think, for example, that in order for this substrate here to bind into the enzyme, I might need some hydrophobic amino acid here, and then you might make this change. But semi-rational design would be you want this substrate to bind, so you think that this amino acid is important, and then you just replace it by all possible amino acids. So you make a combinatorial library of protein variants which are identical except for 1, 2, or 3 locations where you just try out all possible amino acids. And maybe you've heard of directed evolution, which is also commonly used. So in this process, you start with a protein that already has a desired function, and you would like to improve it. So you introduce random mutations, and from the pool of generated variants, you try them, you test them for their function, and you pick out the one that performs best and restart the whole process. So it's accelerated evolution. And this process also got a Nobel Prize a couple of years ago. But all of these, rational design and directed evolution, they are really time-consuming and expensive and are usually only suited to optimize existing proteins, make small changes to make a protein more performant. But the main challenge about these engineering tasks is the incredible size of the search space for optimizing proteins and amino acid sequences. So if you consider that there are 20 naturally occurring amino acids, and each of which can occupy each position in a protein or in an amino acid chain of length 100, then this protein already has 20 to the power of 100 possible sequences. And proteins are usually amino acid chains longer than 100 amino acids, so a couple of hundred up to a few thousand. So yeah, you can see what I mean. And that's where AI comes in and AI can like help to better navigate this search space and like filter it down. And that's now where I will start talking about text-based AI models. And I call this text-based AI models because the inputs to these models are text representations of proteins and small molecules. So I will give a very quick introduction to this. So for proteins, the representation in text is really straightforward because we have the amino acid sequence, and this is like a text representation of the protein. And for machine learning, amino acid sequences, like this one you can see here, are then encoded into some numerical representation, for example, with one-hot encoding or other kind of tokenization, which just each amino acid is treated as a separate token and like words in text processing. And for small molecules, it's a bit more complicated. So what is very often used are in a SMILES code. This is like a linear notation representing the molecular structure using some characters. So atoms are kind of represented by their chemical symbols. Single bonds are omitted, but double bonds are equal signs. And branches are described with parentheses, and rings are like denoted by numbers, like indicate their connection points. So you can see, then you can translate a small molecule like this into like a linear text representation. And for machine learning, these SMILES codes are also like tokenized and mapped to a fixed vocabulary. And you might have noticed it's already like with tokenization and text representations, it sounds like natural language processing. And these methods have really had like a big impact on this field because there is this analogy between like the building blocks of language and those of proteins and small molecules. So in proteins, like in natural language, protein sequences have their own kind of grammar, and they also contain these long-range dependencies. So you can, for example, think of amino acids as words, sequences of amino acids as sentences, and proteins as, for example, a text paragraph. And for small molecules, it's similar. They also are linear sequences that consist of a fixed vocabulary, and there are also rules and patterns of how they are written, basically. So both protein sequences and SMILES codes are compatible with language model architectures designed to handle such linear sequences, and that's why the methods of NLP are very often applied to proteins and small molecules. And at this point, I would like to give you like a first... Oh, there's a question, yes?
How good does tokenization work for these types of problems? Because if you think about NLPs, tokenization can still be a huge problem in some settings. That you don't have exactly these tokens you don't know you want to have to find similarities, for instance.
Yes. So in case of proteins, the library of tokens that you need is really, it's just the 20 possible amino acids, right? But with SMILES, it's a little bit more complicated. But also there, I think they use a fixed vocabulary of tokens, which is not that large, and then they just map these SMILES, like each recognizable group in the SMILES to a token.
I'm just curious because I know if you have an English database, tokenization works quite well. If you take a German database or any other language, tokenization fails typically. So
Yes.
I'm wondering how good tokenization works for if you take SMILES.
I think it works fine, but I would need to look into this more closely. All right. So I would like to give you a first example of a model that has actually been generated by some chemists here at ETH, and it's a transformer model trained to predict the temperature-dependent limiting activity coefficients of molecules. It's not very important right now to know what this is. It's just a property that small molecules have that describes their behavior in a solution with a given solvent. And the input to these models are SMILES of the solute and the SMILES code of the solvent. And the output is basically just the predicted activity coefficient of the solute. So the SMILES codes of the solvent and the solute are tokenized into an input embedding, and the temperature information is added to this input matrix. And then this transformer block transforms this input embedding and feeds the new representation into a regression head that outputs a single value representing this limiting activity coefficient prediction. And this model has been trained on experimental data. So for some molecules, you just know this limiting activity coefficient. So it has been trained in a supervised manner, and they have shown that they can quite well generalize to other molecules that the model has never seen. So this just as a first example how these SMILES can be used for a small molecule, molecular property prediction. And now about protein, you might have heard about ESM. It's also quite famous. It's the model that has been trained by Meta. And it's basically a self-supervised learning model with the masked language model objective on protein sequences. So it was trained on 250 million protein sequences, so a lot. And it works in a way that each input sequence, for example, of this protein here, is corrupted by replacing a small fraction of the amino acids by a mask. And then this masked sequence is encoded in a numerical representation, and this ESM transformer generates a new representation of this input sequence that allows it to make a prediction which amino acids have been masked in the sequence. And if the prediction is correct, so it compares its prediction to what has actually been masked. And if it's not correct, the prediction, then the model is improved. And that's how this model is trained. And it turns out that if this model is consistently performing well on this task of finding a couple of amino acids that have been masked from the sequence, then it needs to know a lot about protein sequences and protein structures also. You can imagine that this is a really simple task, but it's not easy at all. So if you, for example, want to fill a gap in a protein, you need to know a lot about the protein to make a good guess, basically. So you also need to know about the structure of the protein, for example, because some amino acids might actually fit in there and some might not, right? So it turns out that in this hidden state representation that the protein generates, the ESM model generates, there's a lot of intrinsic biological property information and also information about the protein structure. And this is just encoded in this hidden state representation, in this embedding. And that's extremely interesting also to use these representations for downstream applications. And the authors, they showed this also with these orthologous proteins. So they showed that if they take some data set of orthologous genes and proteins, so these are related proteins that come from the same ancestral gene, basically. So orthologous proteins tend to have the same function. And you can see the untrained transformer takes these orthologous genes. Each group of genes is colored in one color, and then the untrained transformer generates a representation like this. If you take the representation and do some dimensionality reduction with t-SNE, for example, you can see that The trained transformer is able to group orthologous proteins and cluster them. So it learns something about the function of these proteins from these 250 million sequences that this is trained on. So this shows that this latent space representation that this model generates are very useful for transfer learning. So you can, for example, fine-tune this language model on a specific downstream task. So you can use supervised learning with a potentially quite small data set that is labeled to fine-tune this model for a particular application, which might be like protein function prediction, prediction of mutational effects, prediction of protein stability, or even the prediction of 3D structure. And the potential of these self-supervised transformer embeddings are currently like starting to be exploited by more and more researchers and models. So now I've already mentioned the protein structure. So I would like to transition to the next topic of protein structure prediction. So first, before I start talking about AlphaFold and again ESM, I would like to just quickly say a few things about protein structure and why it is important. So protein structure, simply said, just refers to the 3D coordinates of all the atoms in the protein in its folded state. So, and knowing this is very useful and important because it gives us like more understanding of a protein, because the structure, as I said earlier, defines the function of the protein. And so knowing the structure helps us to find like the biological role of a protein. And for drug discovery, for example, knowing the protein structure might help us to identify some active sites or some pockets in the protein that we might, for example, target with a small molecule drug. For example, if you have the structure of this protein, and this is a bad protein involved in some disease, you might see that there is some groove here, and that this is a good like target site to target with a small molecule drug, for example. However, for many years, researchers have always relied on very time-consuming methods to generate protein structures, like X-ray crystallography or NMR, to find these structures for a given sequence. But recently published models have, like deep learning models, have made huge progress in predicting structures without these experimental procedures. So quickly about the task of folding a protein and predicting its fold from sequence. This is an extremely difficult and challenging task because these amino acids have a lot of degrees of freedom. They can rotate around their backbone bonds and side chain bonds. So the number of possible structures for even a very small protein is astronomical. And folding is also driven by various different interactions which happen between the side chains of the amino acids, and they are really difficult to model, like hydrogen bonds, hydrophobic interactions, and van der Waals forces. And there are also long-range dependencies. So the folding is influenced by interactions between amino acids that might be really far apart in the sequence of the protein, but then come close together in the 3D fold. So if you use a model to process sequences, it needs to be a model that can handle such long-range dependencies. And finally, the training data is relatively scarce, even though we have been characterizing protein structures with these experimental methods for decades. The number of solved protein structures is still relatively small from a deep learning perspective. So, and for this task, because it's so important, there's also this critical assessment of protein structure prediction competition that takes place every couple of years and invites like researchers and machine learning experts to give a try on predicting folding of proteins from sequences. And this competition was, like the 14th holding of this competition was won by AlphaFold 2, and by quite a large margin. And the predictions of AlphaFold showed a root mean squared deviation of the C alpha atoms, so the tips of these backbone triangles that each amino acid has, of only 0.96 angstroms. So this is the root mean squared deviation of these C alpha atoms. And to compare, like the width of a carbon atom is 1.4 angstroms, so it is quite precise, the prediction of AlphaFold. And you can see here also like an experimental structure in green, like aligned with an AlphaFold prediction, just as an example, how precise AlphaFold can be. And this is really kind of a breakthrough in the field of structural bioinformatics. And that's why I will now dive a little bit deeper into like the architecture of AlphaFold. But still I can't like explain everything because it's quite a complicated model, so I will explain it on quite a high level. But important to know is that the input to the model is basically the input sequence, the protein sequence that you want to fold. And then what the model does, it searches a genetic database for similar sequences, possibly in other organisms, and makes an alignment. And that's what is called a multiple sequence alignments. So it generates this, and then it also looks in a structure database for similar proteins that for which already a structure exists. And using these templates, it generates a first guess kind of the pairwise distances between all amino acids in the protein. So it kind of generates a first prediction of the structure based on these templates and some statistical analysis of like known protein structures. And then from this multiple sequence alignment and this pairwise distance matrix, the model generates this MSA representation and the pair representation. So the MSA representation is just the multiple sequence alignments translated into this representation containing like a lot of sequence conversation information and also about sequence entropy. And the pair representation is, as I said, based on the analysis of known protein structures called templates, and is kind of an initial prediction of the pairwise amino acid distances. And then these two representations, they are passed to the evoformer, which is the core of the AlphaFold architecture. And this transformer model updates these two representations through attention mechanisms and convolutional-like operators, and generates like a new representations with like updated representations. And then these are passed to the structure module, which converts these updated representations into spatial coordinates. So it predicts the distances between all the residue pairs and the angles of bonds in the protein, and from this constructs a 3D model of the protein. And it also applies this interesting concept of recycling that you see more and more often nowadays. So it's kind of an iterative refinement process where the output of the structure module is like fed back to the evoformer multiple times. So if the model has made a first prediction, it's not outputting this prediction yet, but it's giving it back and adds it to the pair and MSA representations and makes another pass of the model. So this helps refine the predictions by allowing the network to reconsider its earlier outputs, basically. And this benefits, like the model benefits of the additional contextual information that it gets from subsequent layers. So they do this recycling 3 times before they output the final structure and the 3D model. Okay. And AlphaFold training is also quite complex. There are several structural objectives. So where deviations from the true structure is like penalized with different loss terms. And in addition to this, there's also like a BERT objective. That's how the authors call it, because they randomly mask a part of the multiple sequence alignment, and the network is then required to like reconstruct the masked regions. And this is also like an additional training objective. And then there's also an interesting principle applied, the training with self-distillation. So they have a certain data set of protein structures that have been resolved with these experimental methods over decades of hard work. And they train their model on this data set of known structures using conventional supervised learning. But then, once the model has achieved a certain level, they use sequences of another 350,000 proteins of which we don't know the structure, and make a prediction using their trained model. And then, once they have made this prediction, they merge the two data sets. They take the experimentally derived structures and their predictions of this already trained model and retrain the model from scratch on this merged data set. And you can repeat this several times, and this apparently makes effective use of like unlabeled sequences of which we don't know the structure. And apparently this really improves the accuracy of the model, which I find very interesting. Good. So just check the time. Maybe, is it all right if we take like a 10-minute break now and continue after? All right, is everybody here? Can we continue? Nice. So before the break, we talked about AlphaFold to predict protein structures. And I would like to remind you of this model here that I showed earlier, the self-supervised model trained on 250 million sequences with this masked language model objective. And this model has been extended, or first the authors, they have shown that in these hidden state representations that are generated for this model, there's also information about the structure of the protein. So the model cannot observe the protein structure directly because it only gets sequences, but the structure of the protein is kind of like a hidden variable that influences the patterns in the sequence. So the model can still learn about the structure of a protein only from the sequences. And they showed this by using a data set of of proteins with known structures, and then added a label to each amino acid of these proteins, like whether they are part of a helix or a strand or a coil, so the type of secondary structure they are in. So you can see this here, like the strands are in green, the helices are in red. And then they derive this transformer hidden representation for all the proteins in their data set, and then they just fit a very simple logistic regression classifier to predict a secondary structure, like to classify for each amino acid in the protein, whether it is part of a helix or of a strand. And they show that with this very simple classifier and using the hidden state representation as input, they can achieve quite high classification accuracy regarding these secondary structure elements. And this shows that this self-supervised training only on sequences also like encodes information about the structure of the protein in the representations of the transformer. And then there has been like a second version of this model released, so ESM2, And they have extended the model with a folding head, and that just extracts like atomic coordinates from the hidden state representations of the ESM model. So they take this ESM model that has been pre-trained on all these 250 million sequences with the masked language model objective, and they fine-tune it and add this folding transformer block here. They also apply this recycling strategy from AlphaFold. And then they train this, they fine-tune the model on 300,000 experimentally determined PDB, like protein structures from the protein data bank. And they achieve prediction accuracy similar to AlphaFold. So this is kind of remarkable because this model removes the most costly aspects of AlphaFold. So it doesn't need to access any databases to look for templates and similar sequences. So it doesn't have to do this multiple sequence alignment. And this makes the model about 60 times faster to predict protein structures compared to AlphaFold. And it's like completely standalone, so it doesn't need external databases. So it just takes sequences and outputs a structure. And if we compare the performance, and we split this comparison into like backbone atoms and side chain atoms. So you remember the backbones are these three atoms, the links of the chain, right? So both models perform quite well to predict the structure, the position of these backbone atoms, but AlphaFold is often a bit better. And regarding the side chain atoms, this is a bit more difficult, so the performance of both models drops because the positions of these side chains are usually harder to predict because they are more flexible. And AlphaFold still generally performs better in positioning these side chains. So if you have an application where high accuracy of backbone atoms and side chains is really crucial, then I would recommend you use AlphaFold. But ESM and ESMFold provides like a really valuable tool if quick predictions are needed across, for example, a vast number of proteins. And just in general, the obtaining of protein structures is a lot easier thanks to such models. But you also have to be careful because they are still only like near experimental accuracy. And also regarding protein dynamics, there's some things you have to like consider, that if a protein is very flexible and has like parts that can move and constantly do this because proteins are dynamic, then from this whole range of dynamic movement of a protein, AlphaFold and ESM will just give you some average conformation basically. So it's a bit difficult. You might, for example, predict the structure of an enzyme of which you know it catalyzes a certain small molecule or substrate, and then you predict the structure of this enzyme, and you find out that in this structure, the small molecule doesn't fit into the active site anymore. And this is because the protein in reality like can move a bit and can like open its active site a bit. And then it can be that your AlphaFold or ESM structure gives you a conformational state that doesn't allow for this substrate to enter the active site. So you have to make you have to think about this, that this can be a problem. So now that we know how to predict protein structures with deep learning, I would like to talk about AI models that use this structural information as input. So I will first talk about 3D convolutional neural networks and then about graph-based models. But before I can do this, I will give a short, not so short introduction to graphs and graph neural networks so that we all have like the basics to understand the models that will follow then. Yes? So if you take a protein that has like a few hundred amino acids, then it might take like 15 minutes would be normal for AlphaFold, for example. But I think with ESM it's less than a minute. So, or for small proteins might be 10 seconds, something like this. All right. So first, I would repeat some concept about the structure of proteins. So the structure of proteins, as I said, determines the function of a protein. And these proteins, they fold into these complex shapes that like allow them to interact with other molecules. And a protein sequence alone provides like no information about the actual arrangement of the atoms in 3D space, right? So models that incorporate not only the sequence of a protein should be superior at tasks that require an understanding of the physical placement of residues. For example, tasks of interaction prediction or enzyme activity prediction. So I guess you agree with me that to make a prediction whether this substrate here can be processed by this enzyme, the information of the physical placement of these residues will be quite, or should be really beneficial for a successful prediction. But there are also like challenges associated to using structural data. So structural data is much less available, so it's limited availability, and sequence-based methods are usually more simple and more computationally efficient. So about 3D CNNs, I will explain just the general procedures and how these can be used to, for example, process proteins. And so if you have a protein structure like this, to convert the atomic coordinates into a format suitable for a 3D CNN, the space around the protein is divided into a grid of so-called voxels. And then you can use different types of atomic properties to define multiple channels in the input data. For example, the types of the atoms. Here in this example, they have a carbon channel and a nitrogen channel and an oxygen channel. And then they use like convolution and pooling to extract the relevant information from these three-dimensional grids. And in the end, if they have reduced it to a single vector, they use a fully connected layer to derive like a global prediction. And such 3D CNNs have been shown to perform well in tasks such as protein stability prediction, for example. And this example is also a model that some people here at ETH have developed. It's a three-dimensional CNN for the prediction of protein ligand affinity prediction. And a ligand refers to a small molecule that binds to a protein. So as you can see here, this small molecule binds, for example, to this protein structure. And the affinity refers to the strength of this interaction. So if this binding is strong or weak. And they want to predict, given an input structure like this, so a protein with a bound small molecule, if this interaction will be of high strength, basically. So what they do is they center a grid around the geometric center of this ligand, and the position of each atom is then mapped to one of the voxels. And they also use additional chemical properties of the atoms and the structures to define additional channels. For example, here they have a channel for aromatic carbon atoms, one for hydrogen bond donors and for something else. And then they use also a convolutional neural network that reduces this 3D grid to a single PKD value, which represents an affinity prediction. And such 3D CNNs were the first structure-based models to outperform sequence-based affinity prediction. okay. But these 3D CNNs also have some drawbacks. So the model sizes are usually much larger than compared to 2D CNNs. The parameters of the models, they grow kind of cubically with the resolution of the grid and the size of the grid. And sparsity is also an issue, because if you, for example, model such molecular interactions, like a portion of the voxels in your grid might just represent empty or homogeneous regions that don't really contribute any meaningful information. And the inefficiency arises because these voxels are still processed by the model. And then rotational invariance is also a problem. So to achieve invariant predictions, regardless of the spatial orientation of the 3D object in the input data, you have to present these objects in multiple orientation, which makes training of the model also a lot more computationally costly. So 3D CNNs usually require large training data sets to perform well and to avoid overfitting. And training and inference require significant memory and processing power. So the application to large data sets of structural data is limited. For example, if you do some molecular dynamic simulation and you want to process like the frames you generate during this simulation, it would be better if you had something that is a bit more efficient. And a potential alternative to these 3D CNNs are graphs, like graph modeling and graph neural networks. And so I'll give you now a short introduction to this before I start talking about graph-based AI models. So graphs describe a set of objects that are then called nodes, and the connections between these objects that are called edges. And information in such a graph can be stored in each node of the graph and also in each edge of the graph. And you can, for example, also have some global context vector, some data that is associated with the graph. And this is a really powerful representation of data, especially for non-Euclidean and unordered data. For example, you can think of a social network with friendships, or citation networks, or also the atoms in a molecule. And graph neural networks are just neural networks that have been developed to process such graph structures. So if we look at this, potentially a social network with some interactions, we can build a graph representing like people as nodes and their relationships as edges, for example. And then if we have this, we can save the connectivity of this graph. So which individuals or nodes are like interacting with other individuals. We can save this connectivity in an adjacency matrix, which is a square matrix with the dimension number of nodes in the graph times number of nodes. And the elements of the matrix indicate whether pairs of nodes are connected or not. So you have either a 1 or a 0 in this matrix. And this tells you that, for example, this person is connected to this person. So a second example would be this caffeine molecule here that you could represent as a graph. And then this is a very convenient and natural abstraction to describe molecules, where atoms are translated to nodes, and the bonds between atoms are translated to edges. And for this molecule, a graph would then look like this, and the adjacency matrix would look like this. Then, to pass such graphs to a neural network, we need to bring this type of information that is associated with a graph into a matrix format. So the node features, the edge features, the connectivity, and also the global context. So what you usually do is you assign each node an index, and then you store the features of these nodes. So the feature of node E in row E of the node feature matrix. So then for the connectivity of the graphs, you have this adjacency matrix, as I explained before, but we usually expect the number of connections to be much lower than the number of entries in this matrix. So these adjacency matrices are usually very sparse, especially for large graphs. And that's why usually this other representation, this edge index, which is kind of an adjacency list, is often used. And it's like a more memory efficient way to represent the connections in the graph without using like memory for saving the unconnected parts of the graph. So you basically have a list of nodes that are connected. So node 2 is connected to node 1, node 3 to node 1, node 3 to node 2, and so on. And this is a much more sparse representation of the connectivity, right? And then if you have this edge index, you can save the edge features in the edge feature matrix in the same order as in the edge index, basically. And then the global context vector, you also just have it in a matrix format, and then you can pass these information to a graph neural network. But first, like what kind of tasks do we want to solve with graph neural networks? And something really common is like a graph level task. So you have an entire graph, and you want to predict a single property for this whole graph. So for example, taking this graph of a molecule, you might want to find out, is this molecule an antibiotic? And then there's node level tasks. So you predict some property for each node in a graph. So this is a super famous example of this toy data set of this karate club, where the two instructors, they have an argument, and they have to split the club. And you have this graph of the social interactions between the members, and the task is to predict like which member will stay loyal to which of the instructors. So it's a node level task because you want to predict something for each node in the graph. And there are also edge level tasks which predict a property or the presence of edges in a graph. So a good example would be like the friendship suggestions on Facebook. So starting with graph neural networks, a GNN or graph neural network is kind of an optimizable transformation on all attributes of the graph. So the nodes are transformed, the edge features are transformed, and the global context vector can be transformed. But it preserves the graph connectivity. So it's this graph in, graph out principle that the model accepts a graph as input with information loaded in the nodes, edges, and the global context vector, and then progressively transforms these embeddings without changing the connectivity of the graph. And if you, for example, want to do binary prediction on the nodes, like for this karate data set, then to get a prediction for each node in the graph, you could, for example, apply a linear classifier to the transformed node embeddings, and then derive a prediction for each node. And the same could also be done for edges if you want to classify some relationships, for example, between the members. And the edge features and the global context information, they can also be included if you, for example, want to make a prediction on the nodes. So to update and transform these features that are saved in the nodes, in the edges and in the global context vector, GNNs usually apply this message passing neural network framework, so that neighboring nodes or edges exchange information between them and influence each other's updated embeddings. And this makes the learned embeddings aware of the graph connectivity and aware of the neighborhood, like of a given node in the graph. And this message passing works in 3 steps. So if you look from the perspective of this node here, you want to update and transform its features. Then you gather all the neighboring node embeddings or messages, like those from these 2 nodes it is connected to, and you aggregate these messages using an aggregation function. Here in this figure, it's just adding them up, and you usually include the node features of the node that is of the node in question, basically. So, and then once you have these aggregated messages, you pass them through an update function, usually a learned neural network, to generate the new node embeddings for this node. And you do the same thing for each node in the graph, collecting messages from other connected nodes, aggregating the messages, and passing them through like a small neural network to generate new node embeddings. The same can be done also for edges. You can also convolve, do message passing from edge to edge. And this message passing process has some similarity to convolution on images, on 2D images. So both are operations that aggregate and process information of an element's neighbors to update the element's value, basically. So in graphs, the elements are neighboring nodes, and in images, the elements are the neighboring pixels. So in graphs, the number of neighboring elements, so the number of neighboring nodes, can be variable, but in an image, this is not the case. So the neighbors in an image is just the adjacent pixels, right? So this is one difference. But you could basically say that image convolution can be thought of a graph convolution on a graph with an ordered structure where each node represents a pixel and is connected to its adjacent pixels. So because of this similarity, this message passing process on graphs is also called graph convolution. And as in 2D images that are processed with CNNs, we can also stack graph convolutional layers. Then if we, for example, have 3 layers, a node after the 3 layers will have information about nodes that are up to 3 steps away from it, basically. Because in each layer, messages are sent like along all edges, and then if you do this 3 times, information will flow along 3 edges, basically. And there are several types of graph convolutional layers that are commonly used, and each has like a different aggregating function to aggregate information from neighboring nodes. So I have shown here just 2 basic ones. So this GCN-conv layer, it updates the nodes, the node features of node E for the next layer, L+1, using this formula here. So for each neighbor of node E, which includes also itself, you take the features of this neighbor, the node features, you multiply them with a learnable weight matrix, and then you normalize with the degrees of the 2 nodes, and you sum this term up for each neighbor of a given node I, and pass this through an activation function to get the new node embeddings for this node I. And this graph attention network layer works similarly, but it also sums over the neighbors, takes the features of the neighbors, multiplies them with a learnable weight matrix, and with an attention score. So you compute an attention score, which refers to like the attention score between this node I and the node J. So, and these attention scores are computed based on the features of the 2 nodes and the edge between them. So this is a convolutional layer with some attention scoring. So from each node's perspective, you basically assign a certain weight to its neighbors. So, and these weights are taken into account in this aggregation function. Good. So I will continue now with the graph-based AI model, give you one or two examples, and hope that this introduction was enough to help you understand these models. So this is a very nice paper from 2020, where they trained a graph neural network to predict whether molecules have an antibiotic effect or not. So it's kind of a graph-level binary classification task. So you generate a graph representation of the molecules in your data set, and then they added also like chemical data to the nodes and the edges. So for each node which represents an atom, they added atomic properties like what type of atom it is, what if it's charged, and this kind of information. And for each edge, they added information about the chemical bond it represents. So like if it's part of a ring, or like if it's a single or a double bond. And then they use these graph representations that are like featurized with this chemical information to train a graph neural network. And the training set in this case was 2,560 drug molecules of like diverse structure and function, and 120 of them were labeled with known growth inhibition of E. coli. So 120 of them are known antibiotics. And then they do very simple graph convolution on these molecules and apply a global pooling. So after the node features and edge features are transformed, they take the features of all nodes and edges and pool them into a single representation and feed this into a neural network that outputs like the predicted probability of growth inhibition of E. coli. And then once they have trained this model, they applied it to a larger library of molecular structures, to the drug repurposing hub with 6,000 molecules. So they did inference on this library of drug molecules to do some classification if some of them might be antibiotics. And it turns out that among the 99 molecules for which the model predicted highest probability, actually 51 of them showed growth inhibition against E. coli. And just to compare, among the 63 molecules that received the lowest probability predicted by the model, only 2 of them displayed growth inhibition. So this is quite a remarkable result, I think. Yes?
So the number of training data on the previous slide was extremely small. How did they avoid overfitting?
Yeah, the question is how they avoid overfitting, given that the training data is, like the training data set is really small. And yeah, I'm not exactly sure like about regularization techniques that they have used in their model architecture, but I know that the architecture of this model, this graph neural network, is also like extremely simple. So it will also have very few parameters, but I don't know by heart how many it is. But yeah, this certainly helps, I think. But yes, given that the data set for training is really small, I think this is quite a remarkable result. And it also led to the discovery of Holicine, which is like a new antibiotic that displays bactericidal effects like against a wide range of different bacteria. And you can see like here what they published in their paper, that you have a certain concentration of bacteria, and if you incubate them with concentrations of this Holicine molecule, you will see that the concentration of bacteria declines, so they die. And then here, they also took the same bacteria and pre-treated them with ampicillin, quite an old antibiotic for which some resisting bacteria strains exist already. And you see that some of the bacteria, they persist after being treated with ampicillin, and they just show that they can even kill these persisters, right? So if they apply this holicin. So it's just a functional antibiotic that has been discovered in a very simple manner, basically. And interesting is also that this new antibiotic has kind of a low structural similarity to other known antibiotics, which suggests that the model was kind of capable to generalize. It didn't just predict molecules that have a similar structure to the antibiotic molecules it has been trained on, but it kind of generalized and accessed somehow new antibiotic chemistry, which I think is quite interesting that this has worked. So then this study has modeled small molecules as graphs, but you can theoretically extend this also to proteins, right? Because proteins are just large molecules with a fixed vocabulary of building blocks. So you can also model a whole protein as a graph, right? And this here is like a graph modeling on atom level, but this study I would like to show uses a kind of different approach, so a more coarse-grained approach. It's a graph convolutional neural network for the prediction of protein functions. So it takes protein structures, and then it models them as a point cloud of amino acids. So it reduces each amino acid to its C-alpha atom, basically, and then connects these points representing the C-alpha atoms of all the amino acids. It connects them if they are less than 10 angstroms apart. And then you get kind of this interconnected graph that shows like the structure of the protein. And it's kind of a contact map, which is the input to this convolutional neural network. So they also featureize these amino acid nodes with embeddings derived from a protein language model. So they can, for example, use embeddings generated by the ESM model and put these embeddings into the node features that represent, like the nodes that represent amino acids. So, and then they have like 3 layers of graph convolution with some skip connections, as you can see here, and a global add pooling. So they again pool the representations, the transformed representations of all the nodes in the graph, and then generate, like with this, a single representation vector, basically, and pass this to a fully connected neural network to derive like probabilities of like class memberships for each protein. So like a prediction to what class the protein belongs, and with this also like what function the protein might have. And this simple GCN outperforms many machine learning tools for protein function classification. And it suggests that the main reason why this model works so well is that this graph modeling that is applied of the protein structures. So there are basically, the convolution sends messages between residues in this graph structure, between residues that are distant in the sequence of the protein, but end up being really close to each other in the 3D structure of the protein. So these residues are then connected because they are close to each other in the 3D structures. So the model has kind of a better awareness of the protein structure, right? And that's hypothesized that this is the main reason that this model has performed better. Yes? Like a feature in the sense of, for example, like a graph level feature. So how, for example, a function of a molecule evolves over time. That doesn't make sense now. I'm not sure actually. So would be interesting to look into. Yeah. So I already reach the summary of this part. So graph neural networks offer many advantages for modeling and processing 3D objects, such as molecules. And they can handle data that is defined in irregular domains without mapping them onto a regular grid. And so graph representations are also invariant to translations and rotations. If you remember that in the 3D CNN you have to present the object in different orientations, but in graph neural networks this is not the case. And it's also a much sparser representation of 3D objects than these voxel grids that we talked about. And there are many possibilities for data integration. You can save data in the nodes, in the edges, in a global context vector, and incorporate this information in the update mechanisms that graph neural networks have. So these graph convolutional neural networks perform well in tasks where like an understanding of the structure of the interconnectivity of the data is important, and they usually perform quite well with relatively few parameters. So to summarize the whole lecture, the text-based AI models, so proteins and small molecules can be represented in text and handled efficiently with methods of natural language processing. And language models trained on these text representations can generate very informative latent space representations and are even able to predict the structure of proteins. And structure-based AI models usually perform better in many tasks where information on the spatial placement of the elements in the data is crucial. And graphs are ideally suited to model and process this kind of non-Euclidean and unordered data, such as molecules and social networks. And as an overall conclusion, like the engineering of proteins and small molecules is very, very challenging because the search space is just incredibly large. And these kind of AI models that I showed you, they can and will probably much more in the future help to navigate these search spaces and accelerate development processes, for example, for new drugs or new functional enzymes. So I suggest that we stop here, and on Friday I will talk about very recent new models about that, like generative AI models for like de novo design of proteins. So without having an actual protein that you change, but you just generate new structures from scratch, for example, with diffusion processes. Yeah, will be interesting. So thanks for your attention, and hopefully see you on Friday.